Dear colleagues, thank you for appreciation of my work. Today I'm pleased to tell you some more on my research experience with hydrated fullerene and tell you some more on biochemical and biophysical properties of hydrated fullerene 60 in a wide range of concentrations. So, as you have already heard, a fullerene is formed of 60 carbon atoms, and it is perfectly symmetrical. The carbon atoms are connected by single and double covalent bonds, and it is hydrophobic. But there is a technology being developed that allows to put a fullerene into a water spherical shell, and in such water, transformed by a fullerene, exhibits peculiar properties quite different from bulk water. There are donor-acceptor interactions between a fullerene surface and oxygen in a molecule of water, and there are traces of these interactions in the absorption spectra of the water modified by a hydrated fullerene. It is shown that hydrated fullerene water exhibits therapeutic properties such as antioxidant, pro-oxidant, anti-age, neuroprotective, hepatoprotective, and various other properties. Here's the overlook how we obtain the solutions of hydrated fullerene. So the producer, Professor Andreevsky, at first in Ukraine, he dissolves fullerene black powder in benzene. The next step is to mix this mixture with water, and then this mixture is sonicated. This allows to obtain cavitation in this mixture, and one fullerene gets included into a spherical shell. And at last, the solvent, the toxic solvent benzene, is removed by vacuum, and finally they obtain this concentrated solution, and then they dissolve it and get this stock solution that we use. The next thing we do is that we carry out successive dilutions that allow us to obtain the concentrations that we work with. So at first we take the stock solution, then we dissolve it 5-fold, then 10-fold, then 100-fold, and so on till we get the concentrations we need. All these manipulations are conducted using purified deionized water, and we use some sort of homeopathic routine, which includes shaking a little bit of these solutions and keeping them in dark places for quite a short period of time. And there is evidence that hydrated fullerene mostly, best of all, works at lower concentrations, not at comparatively high concentrations. The possible mechanism of that is that when there are many molecules of fullerene, they form some sort of a crowd and do not allow each other to form the spherical shell that exhibits these unique properties. And when there are less molecules of hydrated fullerene, then there is enough space for them to work the proper way. And there are various evidence that hydrated fullerene water and exclusion zone water have similarities such as viscosity changes and concentration of a negative charge on the surface. Here's the pictures that depict some of our experiments with hydrated fullerene water and changes in buffer solution of this water compared to pure deionized water. So here we took 2 samples, water and the water modified with hydrated fullerene and containing it at a concentration of 10 to the power negative 17. So we gradually added acid into this water, and we see that in controls, the blue lines, that the pH start to drop gradually, while in case with the presence of hydrated fullerene, then we see that at first water starts to resist these manipulations and doesn't respond quickly to acid addition, but finally they meet in the end. The same situation is with alkali when addition of alkali results in gradual increase of pH and in controls in pure water, and in case of hydrated Fullerene water at a low concentration, it starts to increase not at once, but with some pause. In the next experiment, we carried out tests with pH measurement, but we set it up differently. We took a flask and placed very sensitive pH electrodes in two places of these flasks separately from each other. And you see that in this experiment, with pure water, without Fullerene, that the readings of the two electrodes are similar, so they're very close to each other, and it's okay, and repeated in many repeated experiments. But in the presence of hydrated Fullerene, we see that the readings of the two electrodes differ. There are huger gaps between them, and it shows that there is some heterogeneity in this solution present that we see. This is some illustration of our experiments with proteins that were inactivated by heat. This is the example with alkaline phosphatase. We measured spectroscopic adsorption of the product of the reaction that it carries out, and you see that this line indicates the activity of alkaline phosphatase, which is not inactivated by heat, just ordinary alkaline phosphatase. This is the line 4, is the thermally inactivated control. You see that the activity of the enzyme drops, and line 3 indicates the samples where hydrated Fullerene to an ultra-low concentration was added before heating. So you see that there is some increase compared to the control. And in the presence of hydrated Fullerene at the same concentration after heating, then we see that there is a rather good revival of the enzyme, which is amazing. And the next step we did, we compared the production of the product of the reaction of intact alkaline phosphatase, which is not heated, just the copy of this experiment. The first line, the same here. You see that without hydrated Fullerene, the red line, the line goes one way. But in the presence of hydrated Fullerene, the line goes, repeats this way, and then at the moment when the production of the resulting product of the reaction goes on plateau, then in the presence of hydrated Fullerene, the product continues to be generated. What is interesting that at the same time we measured the temperature in the reactive medium with a very sensitive sensor, and you see that without hydrated Fullerene, the temperature rises or just changes in this way. But in the presence of hydrated Fullerene, the temperature rises slower, and at the final moment, when they begin to differ, the temperature, instead of increasing, goes down. So we can assume that in the presence of hydrated Fullerene, the free energy is not used for temperature rising as without it, but it is used in helping the enzyme to work, so to say. Here's the huge experiment that we carried out, and it was on transformed Chinese hamster cells that were transformed in such a way that they would represent, would work like cancer cells. So they had telomerases that didn't shorten, so to say. And we had 120 flasks. Part of them were controls, part of them were test samples. They all were cultivated for 7 days, and on the 7th day, into controls, we added a portion of water, and in test samples, we added a portion of hydrated Fullerene to gain a final concentration of 10 to the power of negative 19. And you see that addition of hydrated Fullerene immediately resulted in the decrease of the cell population. So it can be interpreted as increased speed of aging of the culture. Then on the 17th day, we did almost the same into the control cells. We added a portion of hydrated fullerene once more. So at first there was water, the second time there was hydrated fullerene. So you see that here on the 17th day there's also a decrease of the population. We also in some samples added a portion of water to be sure. Then we see that just water didn't have any effect on that. And what we did else, we added into the test samples, which were already treated with hydrated fullerene, the fullerene once again. And we see that there is also quite a small, but still a remarkable drop. So this, we can conclude out of that hydrated fullerene may cause, it's quite a paradoxical effect, but this test may show that hydrated fullerene doesn't let proliferate cells that are tumorous uncontrollably. And here is one more, our object. These are tests on blood. These are tests on undiluted human blood. And we have found out that in the process of single electron oxygen reduction and there are energy shifts, and this energy excitation is generated continuously, and it is preceded in human blood. So we added probes for reaction oxygen species, which was lucigenine mostly, and we revealed chemiluminescence from whole undiluted human blood, and we registered it with single photon counter. So this is the sample. This is the sample of one of our tests. We took blood of one person, of single person. We made control. So this is the chemiluminescence of whole blood of this person. So they look alike. But in the presence of hydrated fullerene at these concentrations, you see that these are low concentrations, there are increase in chemiluminescence, which are rather remarkable, so to say. And at some concentrations, there's no reaction. So the concentrations at which we see the biggest effect is 10 to the power of negative 17 and 19, and also negative 6 in this test system. Which is interesting that such reaction is quite individual, and some person's blood may react rather modestly, but in some person's blood there might be huge peaks, much higher than here. And we see that we had an experiment when we took the blood of the same person within a period of 2 months, and we see that this person's blood reacts, we can compare these results. They're comparable. So this is just interesting to notice that it can be repeated even with the same person. And to conclude, I can say that on the one hand, hydrated fullerene in ultra-low homeopathic concentration stabilizes different biological systems such as enzymes, DNA, and cell cultures, and even human undiluted blood. But on the other hand, hydrated fullerene itself leads to the appearance of oscillatory processes, and this may partly explain the increased resistance of the systems to external signals. Thank you so much.